Greetings, guys, gals, and non-binary pals. Welcome back to the Amazing Plant Project. And today we're doing Chapter 3 in the Biology of Plants book, entitled The Plant Cell and the Cell Cycle. Not the entire chapter. It's going to be broken up into two parts. I'm not entirely sure if we're going to wait until next Thursday to do the other part, or if there's a day between now and then that would work for you guys. So, as of right now, it's just today and then the other half next Thursday. We'll see what happens. So, just one sec. I'm gonna, because I couldn't access the stream feed on my phone, so one sec. Need to switch over to this and then share it real quick and that way I actually have it up here in case something's weird with the chat again. Well, I know the audio works. I mean, to be fair, a lot of different accents pop out sometimes. That's one of the usual ones, so I don't know. I mean, I've been to Texas once for like 2 weeks. Don't know where else it would come from, though, to be fair. I don't know. Aside from media. We'll see on that one. Alright, it's like. Come on. Copy. And over here. Stick it in the. If the thing would ever load. Alright, there we go. Come on. Okay. It's like. Okay. Yeah. Okay. When you hit a button and it does something weird. Okay. I mean, yeah, you want your teacups to not have holes in them randomly, which is good. Okay. Oh yeah, make sure that you drink water because it's good and kind of necessary. Alright. Okay, tea has been procured. Awesome. So, let's see if anyone wants to show up for this. Oh, I have an idea. I have a picture that I can put on the Instagram image. That works. Da, 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 da. Okay. Yeah, now we're good. Alright. So, hi everybody. If I can actually get the phone to actually work. There we go. Alright. Now I can see it over there, and I can see it over here, which is good. I can't stream it from the phone, but I can check it. So, there we go. So, we've got some... Where is the start of this chapter? There we go. Alright. So, we've got some ground to cover on here. There are... Right. Let's see, 2, 4, 6, 8, 10, 12, 14. There's 15 different topics in chapter 3. So we're going to do about half of them today. Otherwise, we're going to be here for like 5-6 hours and not having that. No. I think you and me both would have an issue just being there. As much as I like plants, there's a limit. And then you have to let it just kind of percolate in your brain for a while too. So, yeah. Okay. Let me see. Oh, jeez. Ow. Okay. Alright. Hopefully, through the power of a stack of manga, the computer will be slightly more stable-ish. Yeah. At least the base is more stable. The flippy part on the laptop, not so much, but we'll see what happens. Alright. So, the plant cell and the cell cycle. Yes. You need to let it brew and disseminate to all parts of the brain space. Well, I'm glad that it works. Because, yeah. It's still kind of meh, but I'm glad that it works. A little better. Okay. So, cells, as we have previously discussed, are the building blocks of all multi-celled organisms. It's kind of in the name. But the structure and function differs depending on where it is, what kind of organism it is, the age of the organism sometimes, the environment, etc., ad nauseum into the ether of all the things. So we're just going to go through the chapter by points, and like previously I've tried to do like an overview and then terms and whatever, and everything is so connected, and in order to understand the overview you need to understand the terms, so it's all just kind of like bits of information and then terms and things, and it's all kind of in there. So, for the format. And there's no quiz at the end of this one because there's half of another chapter to go for next time. But I'd still recommend taking some notes and stuff for next time and things. Basically, pay attention if you would. So, first we've got cell theory versus organismal theory. Now, cell theory came first, but it's the idea that living organisms are made up of cells, and these cells are of prime importance. Organismal theory, however, is the idea that organisms are made up of cells. Okay, there. That organisms are made up of cells, but the organism itself is of prime importance. And it was likened to the idea that the who was it? I want to say it was Jackson, Andrew Jackson. I can't remember, and it's bugging me now. All right. Jefferson. Okay, there we go. All right. So it was likened to the Jeffersonian model of democracy, where there's the country itself, like the American country, with individual states inside of it, and you would think there's a theory that says both are important, but like, and in that, the states are more important than the country itself, versus organismal theory where the country is the main thing that has some states in it, and the states are important because they're part of the rest of the country. So it's kind of the same of like the individual cells make the whole operate and make everything work, and you can remove, or like there can be an absence of a few cells in a particular section, and yeah, top down versus bottom up, and the plant can be okay. That spot might not be ideal exactly. There might be some structural damage depending on what the cells were doing or what the cells were supposed to be doing for that particular section of the plant's anatomy, but it can work. Versus if the cells themselves can't survive just external from the rest of the plant, essentially, because everything is so interconnected and modulated and stuff internally. So there's that. Caterpillars? Ah, yes. They do damage the plants. And when that kind of predation happens, depending on where it is and the severity, sometimes the plant just sort of expels that part of itself almost. Like if, say, there's a stalk there and there's bites taken out of it that are like half of that particular section, from here up is probably just going to die off. But if something that we're going to get to later, but might as well touch on it now. So if this is the point that the upper part of the plant dies off, then wherever the leaves are, so like say this is where it dies off, but the leaves are here. Like there's some up here and there's some down here. If the leaf is here, that's where the new growth point is. That's where the new apical meristem would be. So if it's just left like that, this whole section is going to rot. So that's one of the reasons that if you've seen my pruning videos and stuff, you want to cut it right above the growth node, because then it can just grow from that. And you want to do it at an angle because any water that hits it, if it's flat, is just going to sit there and decay. But if it's at an angle, it can just run off. And then it's a lot easier for the plant to heal that injury site and grow past that too. I like that. You have been numbed! I disown you and your descendants. Yes. Yeah. It depends on the plant and the environment and the time of year and everything as to whether or not it's more or less of an issue. But if you're deadheading, you want to try and operate with the same kind of awareness, at least. So, especially if it's a time of year that your area is going to get more rain, like dew even does that sometimes. Just be aware that you want, even if it's just like a slight angle, but yeah, because that helps, and then they grow back better and stuff. So good things with that. Okay. So moving on with stuff. So as previously stated, cells are the building blocks of life forms. They're from the smallest organisms that are single-celled to the largest organisms that are several trillion-celled, which that's a lot. But the realization and the idea that organisms are made up of cellular constituent parts was one of the most pivotal advances in the study of living things, as you might expect, because that's what makes up living things. It's a lot easier to understand why the whole might behave in a certain way if you know what the parts are doing. Okay, let me just see if I can adjust this. Okay. There we go. Okay. And it's really kind of remarkable, really. Again, things that get cut out in post that aren't cut out in live. Alright. So, it's kind of remarkable because cells are relatively similar across the board. There's a lot of similarities, basically. But there's also a lot of differences. Aside from just the animal cells don't have a cell wall, plant cells do. Because that's the kind of elementary school distinction for a lot of people, which is still accurate, but not the only thing. So each cell is more or less a self-contained, self-sufficient component of the whole, and operates under the direction of the other signals in the rest of the plant body or animal body, depending on the kind of cell that you're studying. Okay. So, Alright. Okay, so get to that in a sec. But in a short, yeah, it facilitates the communication between cells. Okay, so first we've got the plasma membrane that is also called the plasma lemma or the cell membrane. It controls the passage of materials in and out of the cell, allowing it to kind of differ from its surroundings biochemically and structurally. So it's a designating encompassing section of the cellular body, if you will. And the enclosed cytoplasm, that's the bits in the cell that's not the differentiated nucleus and other things that we'll get into in a second. But the cytoplasm has different structures and either dissolved or suspended molecules, depending on what the molecules are. They could just be kind of there. They could be disseminating throughout. And each cell likewise carries the cellular, because it's a cell, DNA of that particular organism. So that's set up. We're getting into. Okay, so first main big point: prokaryotes versus eukaryotes. Now, pro would be before, eukaryote or karyote. Carrion, there we go. Carrion, or yeah. Carrion would be the Greek for kernel, being, in this case, the nucleus. So prokaryotes are before a nucleus, eukaryotes are after a nucleus. Now that doesn't mean that there's no nucleus type thing in prokaryotes, but it does mean that they don't have the structure that is known as a nucleus. So for modern prokaryotic cells, the only ones that are still existent, as far as we're aware, at least, are archaea and bacteria. And they kind of function the same as the ancestor prokaryotic cells did. So basically the cellular DNA in prokaryotic cells isn't surrounded by a containing membrane or envelope, membranous envelope, whatever you want to call it. Instead, it is a large sort of circular molecule with a variety of proteins attached to it. And phone, what are you doing? Come on. There we go. Okay. Yeah. Yeah. It's kind of a good way to remember that is like prototype. It's before the type, so it's pro or proto. I mean, honestly, it might make more sense if it was protokaryotes than prokaryotes in that capacity, but it still works. And the nucleus type thing in prokaryotic cells is considered it's called a bacterial chromosome in what is called the nucleoid, which is the prokaryotic equivalent of a nucleus. The issue with that is because there's no extra barrier between the central DNA in the nucleus-esque conglomeration that is the nucleoid and the external, like outside the cell, it's easier to penetrate and disrupt that DNA than it is in a cell that has extra barriers, which is why cells evolved to have a nucleus and then the further structures inside of that. Okay, so now for organelles. So organelles are specialized kind of membrane-bound structures that traditionally thought to be kind of absent from prokaryotic cells. There's a few examples when exactly. There's a few examples where there are organelle type structures in prokaryotes, like acidocalcisomes that have been found in several bacteria and are thought to kind of control the interior acidity of the cell. Now, eukaryotic cells have. Okay, yeah. Eukaryotic cells have a double membrane envelope, and it protects the genetic material in the cell. Hence, that's the chromosomes, the genetic material. And the compartmentalization that's possible in eukaryotic cells is largely due to unit membranes, which is kind of visually definable 3-layered membranes consisting of 2 dark layers separated by a lighter layer. Especially when you look at it with an electron microscope, that's how the coloration and density transmits. Then we've got the cell wall that is essentially, Oreo membranes. Then we've got the cell wall that's more or less rigid, sort of. But it's a structured border for the cell in plant cells. Some protists and most prokaryotes. Now, there are not breaks in the cell wall, but kind of pores, if you will, which don't know how well this is going to transmit on the stream, but there we go. So, and a copy of this is also on my Instagram under Ethnobotanicam. So, there is that. And where is it? Okay, so we've got the cell wall here, and right in here are the gaps in the cell wall, the gaps, which the technical name would be primary pit filled with plasmodesmata. And if you think of the cell as a city, like an old walled city, the plasmodesmata essentially function as gates that are more or less open. Like, they've got some guards on them, but if you're bringing in goods or you're coming into work or something and there's nothing wrong, you can go in, you can come out, that's fine. They might restrict how many things, people, whatever kind of analogy you're going with, can go in or come out at any given point because they're trying to keep an equilibrium to make sure that the tension on the inside of the cell and the outside of the cell remains the same, so that there's no disruption in cellular activity. But there is passage as long as the components attempting to cross the barrier are allowed and can fit, because it's not like just a hole in the side of the cell, it's a perforation. So as long as like nutrients, for example, can fit through the perforation, they can enter the cell. And then we've got over here the nucleus and the nucleolus that we haven't quite gotten to yet, but that's like the genetic sector of regular eukaryotic cells. So we just put that over here for a second. All right. Then we've got the protoplasm that, yes, Border Patrol and ice, basically. Then we've got protoplasm that is the contents of the cells. So that is that's the cytoplasm that has all the associated bits and the nucleus. So the protoplasm is everything making up the interior of the cell within that membrane. The cytoplasm includes the distinct membrane-bound entities in there. The organelles, like plastids, mitochondria, like we will discuss in a bit. Systems of membranes, like the endoplasmic reticulum and Golgi apparatus, and non-membranous entities like ribosomes, actin filaments, and microtubules. The rest is kind of a cellular soup called cytosol. That's just kind of like the cytoplasmic matrix. It's sort of a water with other stuff in it that everything else sort of swims around in. So, got yeah. Alright. So, plasma membrane. It delineates the outer edge of the cell cytoplasm. And alright, so we've got the cell wall out here, and if you look, there's a line right on the inside of the cell wall right here. That is the plasma membrane. Oh man, I don't know why my nose is so itchy right now, but it is. It really does. It really does sound like a Lysol competitor, and it's kind of weird. But yeah. Alright. So the plasma membrane mediates transport of substances in and out of the protoplast. Coordinates the synthesis and assembly of cell wall microfibrils, which is making up the cell wall. and detects and facilitates responses to hormonal and environmental signals involved in the control of cell growth and differentiation. So it's kind of the overseer stopgap to like the first and last point of contact before something enters or exits the cell, basically. And then we've got the vacuoles that are liquid-filled cavities found in the cytoplasm of plant cells. And yeah, basically. And one or more are often found in plant cells, while they're rarely found in animal cells. And they seem to exist in higher capacity or higher number. In higher number when the plants are young, and then as the cells develop and stuff and get more structure, the vacuoles sort of fuse into one larger vacuole that can be up to like 90% of the total mass of that cell, which that's this guy over here. And you've got cytoplasmic strands, just because it's part of the cytoplasm, it's in the cytoplasm, so they go around it too. All right. All right. And the vacuoles are delineated by a tonoplast that is a single layer membrane separating that from the rest of the cell. And if you've ever seen microscope depictions of plant cells, a lot of times you see it like streaming through, or there's movement in the cell itself. I realize that doing that is just shaking the lens. Alright. There's movement in the cell itself, and it's just kind of always moving around in there. And that is cytoplasmic streaming, or cyclosis, which be easy sort of to remember because it's cyclosis, like a cyclone that's spinning. There you go. So that is the movement of the cell's cytoplasm. And if it's the movement of the cytoplasm, it's the movement of everything in the cytoplasm. Which, I mean, honestly, all the components are cytoplasm too. So, it's part of the same thing. But the cell's organelles and other substances suspended in the cytosol can often be observed being swept along in an orderly fashion in the moving I suppose it could be likened to Wi-Fi streaming, except without the connection errors most of the time. Alright. And that continues for the life of the cell. It just continues to move around. Or the interior contents continue to move around. And that facilitates transport of materials and necessary components within the cell and between the individual cell and its environment. That works Go for it, man. All kinds of fun things. And going off that, it's not plant related. But if you haven't seen the anime Cells at Work, I would highly suggest that. Because there are certain depictions and things that make biology, and in that case it's human biology, make a lot more sense with how cells and different bodily systems interact. So, yeah, it's pretty awesome. Alright. Okay, so we've moved over to the next big thing, which, yeah. I saw a couple videos that Dr. Mike on the YouTubes did reviewing certain episodes, and it was pretty cool because he was actually really impressed by it. So, yeah, recommending to doctors is a good idea on that one. But yeah, so, importance of the nucleus. It controls cell activity and determines what and when, like what kind of proteins and when they need to. That's not a sentence. Okay. Restart. It controls cellular activity and via determining what type of proteins and when those proteins need to be synthesized. And it stores genetic information and passes it on to daughter cells during cell division. Because it's dividing the cell and the same material is separated into two different things. Yeah, exactly. And then we've got the nuclear genome, that is the total genetic information stored in the nucleus. And the nuclear envelope, that is a pair of membranes that bind the nucleus, that has many circular little pores that are only visible under an electron microscope, but it allows for an exchange of materials between the nucleus and the cytoplasm. And depending on the species and the location and everything else, the pores are somewhere between 30 and 100 nanometers. That could work. Which, if we've got the... So this right in here would be the nuclear envelope, and then the pores are the little gaps right there. Now we come to the endoplasmic reticulum, which we're going to revisit next time, because this is kind of the short form. And yeah, just excuse me. So yeah, it's a complex kind of 3D membrane system that's present in eukaryotic cells, dividing the cytoplasm into compartments and channels. So those particular portions are densely coated with ribosomes, called the rough endoplasmic reticulum, and there's other portions with fewer or no ribosomes, called the smooth endoplasmic reticulum. Thank you. And that's just denoted by they don't have the ribosomes on them. So that I was attempting to synthesize two different images of cellular makeup for this, and I mean the ribosome, no, the endoplasmic reticulum is situated kind of behind the nucleus in this illustration, because that's where it is right there with the ribosomes. Yeah. And then we've got chromatin, which is made up of DNA and carries genetic cellular information, and histone processes, or like the histone processes would be the group of 5 basic proteins associated with the chromosomes of all eukaryotic cells. It's basically what we were working on last week. There's nucleoplasm, that's the nuclear matrix, so it's the stuff in the nucleus that's not the DNA. And chromosomes, which are probably the most readily discussed outside of like science. But we're gonna go into that now. So during the cellular division, right, the chromatin, that's the DNA, becomes progressively more condensed. So usually it's just kind of there. And during cell division, it becomes more and more condensed until it's visible as distinct chromosomes. And so the chromosomes of non-dividing nuclei are attached at one or more sites to the nuclear envelope. So just kind of there. And the DNA content is much higher in eukaryotes than prokaryotes, which it's a more complex organism, so that makes sense. And yeah, okay. I forgot where the other part of that was, so I'll get to that in a sec. And then there's somatic cells. A little bit, yeah. There's a certain amount of potential politics involved in that one. Alright, and then we've got somatic cells that are made up. They are made up of the rest of the body. They make up the rest of the organism that is not the reproductive cells, basically. Different organisms have different numbers of chromosomes in their cells, somatic cells are the cells in the body, basically. Gametes are the sex cells, so just like with humans and other animals, it's still the same principle. And gametes only have half of the DNA, half the chromosome set that the rest of the organism has, because it's needing another half of the DNA strand to create something. So when it's half of that, or a single set of chromosomes, it's considered haploid, and that's pretty much across the board for gametes. And because the base number of chromosomes can kind of differ depending on the species, the haploid number is designated as N. So I mean, they can respond to, there's been studies showing that plants can respond to emotion and sound and things, so I don't know. Could be possible. Right. So haploid is designated as N. So anything that is multiple of that is like 2N, 5N, whatever. So we've got diploid, that's double set of chromosomes found in the rest of the body, essentially. That's designated as 2N because it's got 2 of the chromosome sets. And then we've got polyploid, that are cells that have more than 2 sets of chromosomes. So you've got 3N, 4N, 5N, or higher, depending on the cell and what it's doing. Then we've got the nucleoli, that's the largest and most kind of discernible part of the nucleus. It's awesome visible. Yeah. That would be a rather large bunch of chromosomes. What was I doing? So the nucleoli would be the plural for the nucleolus. So the nucleolus would be right about here, where the genetic material is stored, essentially. And that's the part of the nucleus that's most readily visible. Yeah. It's most readily visible under a light microscope. So the light microscopes are the ones that you usually find in like science classes and stuff, where it's got the light shining on whatever the specimen is to make it more transparent so you can actually see what you're doing and like make it have more contrast and stuff. Okay, apparently I need to really turn that off. There. Okay. So in that structure, you've got high concentrations of RNA and proteins along with large loops of DNA, known as nuclear organizer regions, which, I mean, whoever comes up with these, like, it's descriptive, but, like, I don't know. So that would be so there would be one or more nuclear organizer region in a non-dividing nucleus. So it's that particular is the site of formation for ribosomal DNA or RNA. Sorry, scratch that. Ribosomal RNA. So creating the ribosomes. And then, yeah. One sec. I do though, honestly. It's like there's only a certain number of times when like I've been reading something sciencey and had the thought like this sounds like it works in a bank and is just bored with its life. Not that working in a bank is bad, but like there's a certain stereotype. Alright. Okay, so nuclear organizer region, right? Site of formation of ribosomal subunits that are like large and small, depends, because there's no real distinct sizing for the ribosomes, which are then transferred via the nuclear pores out into the rest of the cytosol, where they're assembled to form ribosomes. So it's like the ingredients, and then it's shoved out a pore, and the rest of the cell kind of puts it together how it needs to be. Then we've got the actual ribosomes themselves that are small particles. They're 17 to 23 nanometers in diameter, consisting of protein and RNA. I can get behind that because water is good. Drink water. I'm all for water-consuming drinking games within reason. Just, equilibrium and stuff is good. And well, there's a lot more protein than RNA in ribosomes. The RNA makes up about 60% of the mass of the ribosome. But yeah, that's also why we're doing it in 2 parts, because percolation into your brain. Important. Otherwise you get like to the end and the good thing is everything is going to be staying up on the channel, and I made a playlist for the chapter episodes, so after the stream's over, this one's gonna end up on there too. So yes. Pee breaks are good. Because you gotta keep everything good. And they're situated on the sites that amino acids are linked together to form proteins. And found both in the free cytosol and attached to the endoplasmic reticulum. The plastids and mitochondria have smaller ribosomes, similar to that of prokaryotes. And their ribosomes are involved in protein synthesis. So for that, we've got polysomes or polyribosomes. So they've just shortened it to polysomes, which are aggregates of ribosomes. So poly, many, ribosomes, many ribosomes, polysomes. There you go. Aggregates of ribosomes where protein synthesis occurs. And often cells that synthesize a lot of protein have a pretty extensive ribosome, or a pretty extensive system of polysome-bearing endoplasmic reticulum, because that's what it is. They're often attached to the outer surface of the nuclear envelope, too. So, like, nuclear envelope, so they'd be around there. And really, the only difference between the ribosomes are the proteins that they're producing at any given point. And we've got plastids that are the cell walls, plastids, and vacuoles are all characteristic components of processes concerned with photosynthesis and storage. So the main varieties of plastids, we've got a chloroplast, that's, the green ones, the chromoplasts, and the leucoplasts. Now, each is bound by a two-membrane envelope and might be further differentiated depending on what it's doing, how it's used, where it is, the age of the plant or the cell that it's in, ad nauseum. And then we've got thylakoids that are a system of differentiated membranes found within plastids, which are kind of like, it's a sac-like sort of membranous structure that is found in cyanobacteria because they are also photosynthetic. I mean, it kind of works, but the dark wizards would be more like orange, so yeah. Chromo. Wait, did I write it down wrong? I might have written it down wrong. No. No. Yep, okay. Yep. Yeah, I guess spoiler, the chromoplasts are the orange ones that make carrots orange. Exactly. Very fashionable. But yeah, thylakoids are kind of a sac-like membranous structure in cyanobacteria, and I turn this off. What is it doing? No. The chromoplasts are the orange ones. Okay. All right. Or not. All right. There we go. I mean, at least I know the audio works. Okay. So, sac-like membranous structure. We'll get past this one at some point. In cyanobacteria, because they also synthesize light to they also photosynthesize. Leave it at that. And chloroplasts of eukaryotic organisms. So in chloroplasts, there's stacks of thylakoids that form grana that we'll get into in a second. And the chlorophylls are found within the thylakoids. And when they're stacked, they kind of end up looking like a stack of coins almost. So the grana, or the granulum for the plural, are structures within chloroplasts that are usually seen as grain granules, hence grana or grain. And you can usually see them with a light microscope, and as a series of stacked thylakoids if you have an electron microscope. The grana contain the chlorophylls and carotenoids and are the sites of the light reactions of photosynthesis. And okay, get back to that one in a second. All right. Then we've got the stroma, that it's more or less a homogeneous matrix in the plastids. So for this, we've got the chloroplasts that are these guys, and the starch grains in there, and the stroma would be the mix of stuff in the chloroplast. Oh man. Ow. I don't know why my nose is so itchy today, but it's like right under there. And it's bugging me so much. Alright, so chloroplasts. We've touched on it before, but the meat of it. Sites of photosynthesis. They contain chlorophylls and carotenoid pigments, but most of the time the chlorophyll is most prominent, but it depends on the species. Chlorophyll is responsible for the green color in plants, and the chloroplasts in general are kind of disc-shaped and 4 to 6 micrometers in diameter. To the point that a square millimeter of a leaf that's green can have about 500,000 chloroplasts. It is very annoying. I'm trying to ignore it, but it's right there. Usually, yeah, but that tends to be more in the petal structure. So the chloroplasts are in the leaves and the stem, like basically where the photosynthesis is happening. Most plants don't have a blue tint to the main body. Some do, and that's an interaction between a couple of different materials, and flavonoids are probably a good part of that. Yeah. You're good. It's fine. But it's basically most of the color is in the flower, and for this, it's in the leaves. It's now just turned into hand exercises, apparently. And usually the chloroplasts are kind of lining the edge of the cell to better photosynthesize. And they can rotate around depending on where the light source is. A little bit, yeah. A little bit. Okay, so that framework thing set up, right? So the thylakoids that are the structures within the chloroplast that would be, let's see, the lines going through here, it'd be that. Get to that in a sec. So the thylakoids of the various grana, or grana thylakoids, are interconnected by the thylakoids that go through the stroma. So the stroma thylakoids, and chlorophyll and carotenoid pigments are kind of embedded in the thylakoid membranes. So the chlorophyll and stuff in there. Yeah. And it's interesting because there's starch that's stored as an energy source. That's the little grain dudes in the chlorophyll. However, it's only really present when plants are photosynthesizing. I mean, plants are pretty tasty, so I'm not surprised. Not one bit. That and, like, just general gardening is pretty awesome, so. Right. Okay. We're good? We're good. All right. Okay. Yeah, so seriously. So the starches are only really there when the plant is photosynthesizing, and once it's stopped, once it's in a dark environment, the plant starts converting the starch into sugar so it can continue doing what it needs to do, even though certain functions are shut down. And once all those starches are used up, it takes like 3 or 4 hours for them to reappear in the cells. And they've done studies, and if a plant is in darkness for about 24 hours, there's no starches left in the cell. So, something to be aware of. And chloroplasts can kind of contain one or more nucleoids, just like bacteria can. So just the genetic material for that microcosm. if you will. It's controlled by the nucleus, but it's made from a combination of nuclear and plastid DNA. And most proteins are synthesized in the cytosol, so the soup bit, and then brought in, though the chloroplast is able to do some synthesis itself. And it can synthesize carbohydrates from sunlight and CO2, and chloroplasts are part of the synthesis process of amino acids and fatty acids, and provides storage for starches. Now we've got chromoplasts, that the shape is kind of variable. They lack chlorophyll, but they have carotenoid pigments and are often responsible for yellow, orange, and red colorations. So sometimes chromoplasts replace chloroplasts as fruits and vegetables ripen, so like tomatoes turning red. And on that, most roses that I've seen have had green thorns, but if you look at older branches, sometimes they're red or kind of orangey, at least at the tips. And it's the same sort of principle as leaves in the fall. Like, yeah, it would be lower starch grains if you harvest stuff at night. It depends on when, because if it's closer to dawn, then it would be less than, like, I don't know, at 10 p.m. or something. But yeah. But it's the same principle for, like, fall leaves turning colors. As the pigments decrease in potency, other latent pigments come out. So even if it's not, like, gonna drop whatever the component is that's turning colors, sometimes that's a variable in that particular part of the plant's anatomy. And then we've got leucoplasts that are kind of, they're the more simplistic of the three. So they don't have interior membranes, and there's some, like, amyloplasts that synthesize starch. Others kind of work with oils and proteins, but they don't do all that much that's specialized, but like they do, but yeah. And then we've got proplastids, or preplastids, that are small, colorless, sort of pale green and undifferentiated, and they exist in the meristematic cells of roots and shoots. So from last week, the meristome would be the growth plates, essentially, of the plant. And in this capacity, it would be the primary meristems. So the shoots and the ends of the roots where it would be growing out. And at that point, it's undifferentiated because it is a growth plate and it's waiting for signals from the plant to tell it what kind of cell it needs to be. And then we've got prolamellar bodies that are kind of a semi-crystalline body found in plastids. And it's interesting because plastids are one of the more variable, shall we say, components of cells. And with a name like that, they are highly plastic components, meaning they are highly adaptive and changeable because they can do multiple things. They can change as they need to. They can adapt. Interesting bits. I mean, if you want to, sure. So, a whole bunch of different species of lemmings, apparently. So, yeah, the prolemar bodies. Okay. Aha! There we go. So, semi-crystalline bodies found in plastids, arrested in development by the absence of light, and composed of tubular membranes. Yeah, exactly. Then we've got the proplastids, or like the seed embryos in this capacity. And then we've got the endoplasts, so the plastids containing promolar bodies. So the proplasts or proplastids become endoplasts, and then when exposed to light, develop into chloroplasts. And the membranes of the promolar bodies develop into thylakoids, as we have discussed ad nauseum previously. All right. So there's a few other bits in here. So now we've got mitochondria. Now, in for the mitochondria, that's the little guys around here, the little circly oval looking dudes. Now, we've got mitochondria, or the singular mitochondrion. It's bound by two membranes. The inner has cristae, or parts of the membrane that kind of fold inward, and it's embedded in a dense matrix. And small particles in that matrix are ribosomes. Now, there's a lot of variability in the length and shape and size and everything of them. Now, the one thing that most folks across the board know from science is mitochondria are the powerhouse of the cell. It's a yes and a no. Because mitochondria are essentially the digestive tract of the cell. They're cellular sites of respiration, so they break down compounds to release energy from organic molecules, converting it into molecules of ATP, which the cells need. And they process what has been created. But they're not forming the energy. They're not creating the energy. They're taking the energy that's locked and unlocking it so that the rest of the cellular components can utilize what has already been created. And there's been a few instances where they found bacteria that seem to be lacking mitochondria. But I haven't really found all that much info on that one. Do some more digging on that and see if I can find anything for next round. Mitochondria are kind of in constant, basically, distributing the trust funds to everyone that needs them. They're in constant motion and congregate where energy is required, and often that's spaces where there's high cellular activity, whether it's in single-celled organisms where the flagella are moving around, and we'll get to that later, or spots that have a high energetic turnover in the plant that need, or animal, that are like constantly distributing or exchanging materials because that part of the cell needs more energy. And it's thought that mitochondria may have, or probably at this point, were originally bacteria that weren't fully absorbed when the ancient cells that tried to eat them just kind of nommed them up. So they would still be inside the cell, existing as they were, and it ended up being a I'm blanking on the word for mutually beneficial at this point, but let me see. Symbiotic. There we go. Yeah. Yeah. Indeed they do. So, symbiotic relationship between the main host cell and what would become the mitochondria. And that allowed for protection in environmental extremes, and because of that, eukaryotes were able to kind of make headway into land-dwelling beings and more acidic waters too. Because in situations like that, prokaryotic cells can't really function. Prokaryotic bacteria aren't there, but green algae that is eukaryotic can function, which is pretty cool. And it is descended, or it is descended. It's passed down matrilineally, so the RNA is passed down on the maternal line, which that's why when you do like DNA tests and stuff like that, if you're looking at genetic ancestry, a lot of times it's going by, especially if it's going by blood samples, it's going from the maternal mitochondrial DNA. All right. So we've got Peroxisomes now, or microbodies. So they're spherical organelles bound by a single membrane and range in diameter from like 0.5 to 1.5 micrometers. They're granular, sometimes crystalline, in the interior and composed of protein. There's no internal membranes, and they're self-replicating organelles, but with close ties to the endoplasmic reticulum. They don't have any DNA or ribosomes and have to import all their replication data in order to replicate. Sometimes they play a role in photosynthesis and are kind of closely tied to the mitochondria and chloroplasts in green leaves. And okay, now we've got the vacuoles, and that'll be the last one for today, and we'll pick up with the endoplasmic reticulum next time. So if you want to see a sooner round of plantness, let me know. Otherwise, round 2 or chapter 3 part 2 is going to be next Thursday. So for vacuoles, it's again bound by a single membraned structure, or the tonoplast, the vacuolar membrane, as we kind of touched on before. Most come from the Golgi apparatus. Yeah, just a little bit more. There's half a page of notes. Most come from the Golgi apparatus, but some might come from the endoplasmic reticulum too. And again, we'll touch on the Golgi apparatus next time. Many vacuoles are filled with a liquid, sort of commonly called cell soup. It's mostly water, plus some other components, depending on the species and location and the cell and, whatever. It often contains sugars, organic acids, and amino acids, and sometimes crystals form if there's something in a high enough concentration. Often the immature cell has several vacuoles that kind of increase in size and fuse into a single larger vacuole. And it's more energy efficient to have a large area between the cell's external and internal environment. And it's usually kind of a fail-safe at that point too. Often there's pigment in that section, and it's involved in the breakdown of macromolecules, so the big molecules that need to kind of get broken down in order to actually be usable by the cell. Indeed it is. Indeed it is. So, as a closing bit, the three big parts that define plant cells, right? Plastids, cell wall, and vacuole. So, if there's probably some that are kind of hit or miss on one of those three, but in general, plastids, cell wall, and vacuoles. So, we'll pick up with the endoplasmic reticulum next time. I'm going to try and have new videos out for other topics. I'm going to attempt to start posting stuff as of next week for the rest of the series, because I've got Botany Bites that ideally is Monday, Ethnobotanime or something else that's kind of analysis related on Wednesday, this on Thursday, and then I'm bringing the podcast back, and hopefully that's going to be out on Fridays or Saturdays if there's an issue with Fridays. So there's that. It's either going to be starting next week or the week after, depending on things and such. We'll see. And there's going to be advanced viewing available on my Patreon and sponsors, depending on which one you want to do. I'm still testing stuff out with that, and all the links are going to be in the description down below. If you're watching this after it's aired, there's going to be links in the iCard, probably over there, for anything that I can put in the iCard. Everything else is in the description. There's an image of this guy on my Instagram that is also linked down below, or you can just look up Ethnobotanicam, and it's the one from today. So there we go. Any particular suggestions or recommends, if that's a word, recommendations, just pop them in the comments down below or on the various socials. And yeah, the shortest of the three so far, but pretty chock full of information and stuff, so that works. Yeah, anyone have questions before I cut the stream for today? And yeah, just executive decision. Thursday. We're doing Thursday for stuff. So anyway, any questions before I sign off? All right. In that case, I will see you next time. Have a fabulous rest of the day, and yeah. Oh yeah. If you want to check out not plant things, there's links to my non-plant stuff in the description too. I do a lot of art stuff on the YouTubes and various other things and stuff. And yeah, so have a good one. I will see you next time. And yeah, bye everybody. What? Okay, that was a fail. Okay. All right. Here we go. And end. Yes, I want it. There we go. Bye.